Hi, my name is DeChristopher, and I'm presenting my research entitled An In Silico Approach to Identifying and Evaluating Antibodies to Target SARS-CoV-2 Spike Glycoprotein. So just a brief introduction. SARS-CoV-2 is a deadly virus with no known therapeutic options for patients. I think that's pretty well known. There's a lot of different things in clinical trials, from antivirals to monoclonal antibodies to vaccines. As of yet, nothing really seems to be that effective so far. So we were interested in finding naturally occurring monoclonal antibodies that could neutralize this virus. So how do you find an antibody? An antibody is a Y-shaped molecule, and it works because it binds to an antigen. So it binds to either a protein or peptides from a foreign invader. And antibodies work because they have the ability to remember what an antigen looks like. And this immunological memory has to be stored somewhere, and it inevitably gets stored in an organism's genes. So we looked at our own and mouse genes that code for antibodies to find any antibodies that may be able to recognize the spike glycoprotein of SARS-CoV-2 or COVID-19 and subsequently bind to it, neutralizing it. So I think it's probably important here to say why the spike glycoprotein. I think everyone's probably seen this image of the virus. I kind of like to think of the virus structurally as maybe a tennis ball with spikes coming out of it, and the spikes are what act as a molecular key. So they bind to the ACE2 receptor in the respiratory tract, and that seems to be the main way that the virus gets into a host's body. The ACE2 receptor, angiotensin converting enzyme 2, is a really important receptor within your body. It is involved in regulating blood pressure. So it's really important. It's really obviously not good if there's a virus that's targeting this and potentially inhibiting your body's ability to regulate this function. So we started out in February with a protein sequence of the spike glycoprotein of the virus. So we took this protein sequence and we wanted to search through genes, so we had to convert this sequence written in amino acids into a sequence that would be written in DNA base pairs. So we used this tool called Sequence Manipulation Suite to convert it, and then once we had converted, we used the National Institute of Health's IgG Blast tool, and this is a really great tool that allowed us to search for similarities between the DNA of the spike glycoprotein and the genes in the human and mouse genomes. So this allowed us to identify these genes, and they had significant similarities to the spike glycoprotein, but obviously a gene is not an antibody. So we had to do further work to understand which antibodies these genes coded for. And we eventually were able to find 2 antibodies that these genes coded for, and then we were able to figure out if these 2 antibodies themselves had significant similarities to the SARS-CoV-2 spike glycoprotein, which you can see here that they did. Once we had these 2 antibodies, 8FAB and 3UBX, 8FAB is a human antibody and 3UBX is a mouse antibody, that wasn't enough to say just because we think that this antibody can probably recognize this antigen, that's not enough to necessarily say that it will bind or how well it will bind, or ultimately that's not enough to measure its therapeutic effectiveness. So to try to gauge that, we used a tool called ClusPro, which is a server that's really good at finding the near-native structure of how 2 proteins will interact. So it's really good at figuring out what kind of shapes and how will 2 proteins interact. That's all it can do. It doesn't tell you necessarily the binding affinity. To do that, we took the models, the interaction models generated by ClusPro, and we used this tool called Prodigy, which then can tell us, based on the interfacial contacts between the 2 proteins, what is the likely binding affinity. And ultimately 8-FAB and 3-UBX seem that they will bind with a low binding affinity to the SARS-CoV-2 spike glycoprotein. So it seems to be in the micromolar range. This is orders of magnitude away from a high binding affinity, but there does still seem like there is a low binding affinity. We don't know if this is just something we're seeing in our simulations or if it will actually translate into real life. The tools that we used are pretty accurate, but the novelty of this could also have made them less accurate. We won't know until we do some sort of laboratory test, like an ELISA, say, or immunohistochemistry test, to really understand that. Unfortunately, we can't do that now because of the contagious nature of the virus, but that is something we'd like to do. Even if these two antibodies do have a low binding affinity, we are interested in using them as templates to maybe try to mutate the chains and see if we can, from that, increase the binding affinity. And just one last thing to touch upon in conclusion is what happens if there is a mutation. So right now you have a lot of work being done targeting the spike glycoprotein. So you have a lot of antibody therapies being developed. You have a lot of vaccines that are trying to create antibodies within the body to target the spike glycoprotein. And it feels important to talk about what happens when all of these therapies targeting it come on the market. Will the spike glycoprotein mutate? There was some recent work in Nature that showed how an antibody that targets SARS does not target SARS-CoV-2 just because of one amino acid change to the spike glycoprotein. So we may develop all these different therapies, and that may cause sort of evolutionary stress in the virus, causing it to change in a way that makes many of them no longer effective. So I think it's really important to develop a diversity of approaches to be working on a wide range of different therapies to target the virus so that we can ultimately stop COVID-19. Thank you for your time.